I'm so tiny. Oh, hello. I gotta adjust me. I gotta adjust myself here. Hi. Oh, hi, Weetabix, and thank you for checking in, Weetabix. Hi. I'm not really late, but I thought I was gonna be way more late. I think I was only like a minute off of starting time, so it's not that bad. Or a few minutes off, I guess, because it's 6:09 and I waited for the ads to finish. Okay, but anyway, hi. I got so caught up at just as the time was going to hit exactly the hour. I started monitoring my microphone quality, and I started hearing a crackling noise every time I spoke, and I was like, what the heck is that? That's weird. And then I was like, okay, well, now I have to play with the filters. Like, maybe something's wrong with one of my OBS filters. And then as I started taking things off, like noise suppression, all I started hearing was like a loud crackling noise that's being noise suppressed. I'm like, whoa, that's not right. That's really bad. Why is it crackling? And it's like, oh gosh, why is everything falling apart? Okay, how do I fix this? How do I fix this like right now? And then I quickly learned something's wrong with my XLR cable, or I don't think anything's wrong with it. Like maybe this is just how it is. But it was touching another cable, and it was not happy that it was touching another cable, and so I quickly removed the other cable from the premises, and then it stopped crackling. At least it stopped crackling as much. So yeah, maybe that's like a cheap cable thing. I'm not very like audio technical, like savvy, but it was worrying me like how loud the cable sounds like every time I touch it. And it's like touching little like things like a cup. I think I picked like the cheapest Amazon basic XLR cable, and it's not very well shielded. Yeah, like not shielded enough, as I'm guessing the problem. And I'm guessing like, if you buy a more expensive cable, it's probably like, nicely wrapped, like, those like coil wrapped ones and whatever. Yeah, this one's like the most, I think it was like $20. So, but it's like good enough, but I think it is kind of annoying. I have to like go like, okay, other cables, get out of here. Get. Don't touch the XLR cable, you guys. My desk isn't big enough for all this like shooing things away from it. Maybe this is also like a sign like I need to do like proper cable management, because I clearly I'm having a hard time figuring out how to make space for everything. I need to like move my extension, like my power bricks and stuff. They're just sitting on my desk because I don't have a really good way for them to not be on my desk, because it's like all the cables, like if I put it on the floor, some of the cables are too short to like make it from the floor to where it needs to go. So I have to maybe I just need to invest in a better desk. But desks are so expensive. I've been looking to try to get a standing desk because I keep seeing so many things about sitting is killing us, sitting is killing you, sitting is killing me, sitting is killing everyone, stop sitting all day. I'm just like, oh my gosh, I'm gonna die from sitting all day. Someone help me, I'm gonna die from sitting. And I know it's not actually as serious as that. I think every time I look at the papers, they're looking at a pretty sussy like demographic. Like it's like, oh man, the 7-year-olds are sitting all day and their white matter is just like getting ruined. Oh, thank you for checking in, Rico, and oh, thank you for subscribing again. Thank you. Oh, that reminds me, I am close to finishing some alert animations to make those things more exciting, and I want to share them, but I'll wait till they're done. I'll wait till it's done. I will till they're done. Yeah, thank you for resubscribing. Sometimes I use something like a man strip to attach the bricks to the underside of my desk. Yeah, that's like a good idea. I don't know. I really like the desk I have. I'll post a picture maybe of my setup and post it on the Discord or Twitter. I don't know, either one. And it's like a really cute pink L-shaped desk, and I really like it. But my God, is it the most cheap particle board desk? It's the cheapest like particle board, and like it was a pain to put together. It's not very well built together. It feels like it's going to like fall apart any moment. It's not really going to fall apart, but when I put it together, it did feel like it was going to fall apart. I should invest with my like big girl salary money, like a quality desk. But I went for the aesthetic desk, and it was a mistake. Don't go for the aesthetic desk. It's like going for the aesthetic chair. Like, you know, like the gaming chairs that are like, all, ooh, like, look, it's a collab with like Genshin Impact. Ooh, secret lab gaming chair. I did like the equivalent, but for a table, like, or a desk. I'm the clown. A big standing desk will be nice. Like, and then it's also like, I have plans on moving soon. At least in like a year or two, and then I'm like, oh my God, moving a giant standing desk. It's, I don't know if it's just sitting that is a bad thing as much as being static for a long period of time. I think when I like stop, when I like, you know, actually think about the papers and read the papers, maybe that'll be a good paper discussion we can have about the dangers of sitting. Quote unquote dangers of sitting. I'm not a healthcare professional though, so it's a little bit out of my depth, but it'd just be cool to read together. It is more about being static because I don't think there's even like a beneficial effect of also being like just standing. I think you would have to also like be moving. You have to be like active and like doing something. So maybe I have to like be on a walking pad for the rest of my life. Is that the only way I can be healthy, people? I have to like be permanently on a treadmill power my everything, and then like my brain matter won't be destroyed. Yeah, there was like a recent paper. I got distracted. There was a recent paper where they were looking at how even if you're active, like 60 minutes of activity a week, your brain matter was still getting destroyed, dismantled, whatever, if you sat too long. But then I read the paper and it's like they're 7-year-olds, and I'm like, well, duh, of course their brain matter is getting deteriorated. They're old. I think it's kind of over for them no matter what. Oh, hi, Pi. A long time ago, my high school senpai said that live faces were up regular while standing, so standing equal losing fat. But I think this was just his way to get us to stand during an acapella practice. And that's pretty funny. Yeah, I heard like there's the problem with standing is I think there's like also like if you stand too long, you're dying. That's why there's so many. But then there's so many studies saying standing too long equal bad. But then I think they usually look at people whose main job is standing all day, and then there's obviously a host of like biases related to a job, someone who works a job where they have to stand all day. Like, I think the quality of life when your job is just being a grocery store minimum wage worker that has to stand all day is clearly very different, and it comes with its own unique set of problems that might be hard to like tease out statistically from someone who might have a sitting job. Time for me to get ready to go to work. I don't wanna. Oh, good luck with work. You can do it. You can do it. Today I feel like I super slacked off at work. Like, okay, my usual work, I try to like, I take like a little afternoon nap. I take a little afternoon nap in like at like 2 p.m. most days, like 30 minutes usually. Nothing's going on. Most people in my work do it, so I go Honk shoo, me for 30 minutes. Today, I think I forgot to like set an alarm, and I like woke up and it was 5 p.m. and I was like, oh, I missed the whole afternoon of work. No one's gonna get mad at me or anything because it's not like there's any meetings on Fridays. But I might have to like look. I might have to go review some emails later and to see if I didn't miss anything. It was a little too much of a nap. I was so sleepy. I say that, but after talking about minimum wage standing up all day job, I feel bad. My work location is slow AF. I just watch Pokemon and play Pokemon ROMs on my phone. Yeah, like I think there's obviously like drawbacks still on both. Even if like the minimum wage workers get the stand they have it rough, it's still usually not fun. Doesn't make your job feel not like any better, you know. But Pokemon ROMs all day on your and YouTube is at least kind of nice. I've been playing a lot of Pokemon ROMs on my phone during bed. It's usually during bed. Like I'll get ready for bed and like I'll stay up for like an hour or 2 or 3 and like play Pokemon. I've been playing Pokemon Run and Bun, the Emerald mod. And I've been doing pretty good, but then I've also like kind of cheated. It's not really much of a hardcore Nuzlocke anymore. I'm just simply like I'm save scumming. I'm sorry, I'm save scumming because I don't want to pull up my laptop and see what I'm supposed to anticipate. I just save and I go, okay, that's what I'm gonna fight. Okay, and then if I die, I go like, okay, well, back at it again. I do accept if my Pokemon die during a battle, I will put them away. But I do save scum so I kind of can get a feel of what the next fight is. Or else I would just be at the first gym forever with this mod. The biggest downside right now, I'm in the middle of nowhere. I live on the second floor of my work location, so I never feel like, oh, that is kind of rough. Oh, yeah, I can see how that might, like, be a problem. I've had that problem for a while with work from home. And even now, like even after like a few years of doing it, I feel like I've been considering like I really need to get like an office space. Cause currently, as I was saying, I work on like an L-shaped desk, and one side is for my work, and the other side is also for my personal and streaming. So right now this desk is like functioning as three units in one. This desk can do it all, baby. But it's also like causing like I feel like brain problems. Like it's not real, but it's like this feeling of like, my gosh, I turn to the right and there's my work laptop and work desk and work notes and everything. There's a giant textbook of the WHO guide of hematological malignancies sitting right next to me. And it's not like I'm stressed by my work or anything, but it's just the feeling of like the hematological malignancies are staring at me. I could like sign in on at any time and start doing work. And I used to have that problem. I would get bored doing something and then I would start working for free. I would like open up my laptop and go like, whatever, let me open up a spreadsheet and do some work. I stopped doing that nowadays because I have something else filling up my free time, which is being a VTuber. But yeah, it was a problem. But now it's more like I hate being stared at by my work notes. I was working for free. I can't believe it. I can't believe I would have. It was so bad to the point where even my like manager would be like, "I saw that you edited something at like 9 p.m. on our platform. Don't do that." I was like, "I'm sorry." I was like, I had a thought about KRAS mutations, and I needed to like go do that. I needed to go get that done. And I really needed to write that description. I really needed to write that information that I had in my brain. They're very, at least, you know, I'm very glad that they were my managers are like very healthy and kind. They really look out for my work-life balance. KRAS mutations. There is, I think Yeah, it was like this week there was a nice FDA approval this week on KRAS mutations for low-grade serous ovarian cancer. For any KRAS mutation, go go cancer therapeutic development. Woo! Though I think that indication is going to eventually just be KRAS wild type as well. So it's not like because I think that's what they were originally pushing for, but for now, woo, KRAS mutations. I'm really big on like biomarker-based cancer therapeutics. That's kind of my whole thing right now. I'm a bacteria girl, I know, but like my whole knowledge base in my brain right now is filled with biomarker cancer therapeutics. Anyway, I think I've been like we should start the tier list. I've just been yapping for like 20 minutes. I forgot what we're here for. Okay, let me open up the thing. Where is it? Is it this? Oh, yeah. Now I'm too big. Hold on. I'm covering everything. Okay. I had more things planned for this, but I feel like this will be suffice. This will likely be long enough already with the amount of complaining I'll do about each of these. And at first, if you can't tell, at first this was more about experimental protocols, and then it turned into like steps of experimental protocols because there's kind of like a lot of overlap because a lot of these you're doing multiple experiments to get to that point. Like, you know, to do facts, you have to do like pipetting and you have to do like some kind of purification or whatever. You know. It's but I just was thinking more about how did I feel about doing these specific steps in experimental protocols, and then also it turned into how did I feel about doing anything in lab? Like when I thought about lab, I had to do this thing. How did I feel about it? So we have my S tier, which is I love it. Thank you. I don't know how many we're going to have in here, but And then we have the next tier, A tier. I enjoyed it and did not dread waking up in the morning. This is a high compliment. This is a high compliment for me when it regards to lab. In regards to lab related duties, this is a high compliment. I am neutral. This is also a high compliment. I feel like I am neutral is good. Neutrality is good here. I didn't dread it. I didn't really enjoy doing it, but I didn't hate it. And I didn't cry about doing it. So, it's pretty good. Crying in the morning, yeah, this is where we're like going downhill. Like, if I had to plan out my experimental days, or my weeks usually, it was usually on a weekly planner, and I had this on my to-do list, I would cry. That's this like it would be like I'm crying. I wake up and I go, "Oh my god, it's another day. It's another day of this. I need to leave." Imagine doing anything in lab. Yeah. And that's why I'm not in lab. This is why I don't do any lab work anymore. And will implode if I had to do this again. So this is like the category is worst of the worst of the worst. The thought of doing this again will make me like literally die. D word, unalive, whatever. I will like pass away if I thought, and I would also be complaining. Maybe that would be more of it. I'm like complaining so much I turn into a little rage ball and like implode upon myself. I have some thoughts on some of these. Oh yeah. Well, I'm sure some people have some thoughts. I also like I know some of these might be like quite specific. Like I think some people like gavaging. Gavaging is definitely something I don't expect a whole lot of people had experience with. And lung lavaging. Lung lavaging is also something I don't think most people had experiences. So many words I don't know. Oh, don't worry. I will go through all of these like and explain them too. Don't worry. I'll make sure to explain it so we're all on the same page of why I hate this or why I like it. But it's more likely why I hate it. And I did way more in my like bench scientist years, but my brain really was like struggling to remember specifics. I either like blacked out like on what I did, or it was just so minuscule that I can't remember at all what was going on. Especially like a lot of these are things I did in my like it was in my most recent lab experience, which was in grad, like my thesis lab that I inevitably dropped out of. And I was trying to think of things I did when I was like in more cellular culture labs and like bacterial labs. But honestly, for the life of me, I can't really remember what I did in those labs. It feels like a lifetime ago. And then I was like thinking, oh my gosh, it has been kind of a long time since I did any of those things. So maybe that's why I don't remember. And some of these are pretty general, and I could have like expanded them to like something even more general, like drug dilution. I think drug dilution is more like lab math. I should have said it's more like lab math. And I was gonna put lab math in here, but I thought, I can that'll get covered in that one. Okay, so why don't we get started? And we're starting off real strong here with retroorbital blood collection. Okay. Let me explain first what retroorbital blood collection is, because maybe you can't tell from the picture. So this is when you need to do the experiment, when you need to collect quite a large amount of blood, like blood sample from your mice. So what you would do is you would take I really, really hated this technique. You have to anesthetize, make your mouse go to sleep, and then you have to kind of restrain it and kind of gently like grab onto the mouse so its eye is just slightly bulging out, and then you take your capillary tube, which is like a really thin rod with a hollow inside, and then you need to insert it right in the corner of the mouse's eye. And that's where like the retroorbital sinus is. And like as you gently rotate it, all of this blood just comes out. And it's really, it's just so I hated doing this so much. It was so like I felt so bad for the mouse. It was just no one was happy. The mouse wasn't happy. I wasn't happy. But unfortunately, it's like the best way to like collect a lot of blood. And it is like, and learning how to do it was really hard on me because it's not like messing up is really, really bad. I don't know how else to explain it, but like messing up is really bad. And the thought of like, using mice just to practice on this, it's not fun. Because it's like, yeah, I won't get into how many mice had to be used for testing for me to practice this technique, but it was unfortunately a lot because I was really hesitant on like putting the capillary tube into their little eye area. I mean, it's not going right into their eye, but it's like going right there. And then like I start thinking like, oh my gosh, what if someone did that to me? I'd freak out. Obviously, they're not freaking out because they can't feel a damn thing. They're knocked out. Oh, it's just the worst. This was an experiment where I had to like straight up tell my PI, I was like, I can't do this. I think I did it like a few times like after learning, and I was like, I can't do this anymore. I will not do this anymore. And he was not very happy because it was kind of important to collect the blood samples, especially when we're looking at aging. That was like my big thesis project, is looking at aging, different aging biomarkers, and we wanted to see if there were some certain biomarkers in the blood, and we opted to do other things instead. And he was not happy. And okay, so overall, this is definitely I would implode if I had to do this again. I think, like, it's just, there were, I'm losing it just thinking about the thought of, like, having to do that again. I think also, just seeing the amount of blood that gets collected is quite something. It's kind of nauseating. It's not like a huge amount, because I mean they're little guys, you know. It's not like you're getting a whole lot from them, but it was kind of like gross. But when I told my professor we didn't want to do this again, we did instead opt for, I'll just bring this one up, tail blood collection. This is like a method that was a lot easier on my mental health. This was a lot easier on my mental health, but I still despise doing this. But this wasn't because of how hard it was. It was just, this was so annoying to do. And it was also like, okay, as you can imagine, like I said, the mice, they're little guys. They don't have a whole lot of blood. So, tail blood collection is like, you just make little nicks onto their tail and try to collect the little bits of blood that come out from that. And you want to, like, make the nicks not, like, you want to make them small enough that, like, they're not doing too much damage to the mouse, but deep enough that, the blood is actually coming out of it. And this is not a great method, really, for blood collection, especially in the case that we had to do for experiment, because a lot of times it was just not enough blood. There's not a whole lot of blood coming out of here, especially with the tinier mice. I think the bigger, like, older mice, there's, like, quite a bit of blood that came out. But the tiny mice, they were just, like, nothing. There was, like, nothing coming out. So the problem would be, I would keep nicking them, and I need to collect more blood, and I would nick again, and I would nick again, and I would nick again, and the tail is like covered in scars, and this was, we should not have been doing this. I don't think Cook would have been happy, which is like the animal safety and officers. They are the ones overseeing to make sure you're not doing any unnecessary harm to the animal. I'm sure realistically we probably should have just been doing the retroorbital blood collection, because we shouldn't be covering this little mousey in like a bunch of scars on their tail trying to get just a little ounce of blood. I think I just sucked at this. I'm sure with someone that was better could do this way easier than I. And I know, actually, that's completely true, because I remember watching somebody in my lab who was way more, like, skilled at hands-on experiments than I. I watched her collect blood so easily from the mouse tail, and I was like, oh, okay. And then when I do it, the blood just never actually let properly. And I have no idea why. To this day, I don't know why. But anyway, you could tell when it was a mouse that I was doing tail blood collection on, because their tail looked beat up. Their tail was covered in scars. I was just like, I'm so sorry. Here's a treat, little mousey. I'm not a I think me and mice had a mutual dislike for each other. I didn't give them the love and respect, and they didn't care to do anything for me. We weren't meant to be, is what I think. it came down to. So tail blood collection, even though I sucked at this, it wasn't a crying in the morning. And if I had to do this again, I would be frustrated, but not like imploded. I wouldn't implode upon myself at the thought of doing this again. I'd be like, oh my gosh, really? Really? I wouldn't cry in the morning though. I think I would say it's neutral. I just get frustrated that I'm not very good at it. And it was like the most frustrating because I feel like I did it for so long too. I did it for like a year and I just never got better. There's so many, maybe I'll just go through mouse things because I'm thinking about mouse. Cold room experiments. Oh, this is a crying in the morning for sure. So cold room experiments. So in regards to this is highly specific to my PhD thesis. For my PhD thesis, we were looking at adipose tissue and how it changes when you age. And one of the things we wanted to see was how I'm trying to explain it in simple terms. We would look at young mice and old mice, and we wanted to see how they were able to maintain their body temperature in response to cold, and see if we can like have a young mouse mimic like the body temperature retention of an old mice through pharmacological intervention, aka like if we give it certain therapeutics and drugs that change the expression of certain like markers and cells, will they change more into like an old mouse like expression? Will they be more like a old mouse in regards to how they handle cold? I hated doing these. This is definitely a crying in the morning. This is definitely a crying in the morning aspect. I would like think about this like because we have to plan a lot of these experiments quite a bit in advance. You know, you discuss with your PI, your lab guy, and you go, "Hey, we should try to get this kind of data." And then they go, "Oh, we should do this experiment." And so, you know, you have this in your schedule and calendar. And I would see it's cold room time. It's over. It was like it was not even just crying in the morning. It was probably like crying like for the first few mornings like leading up to that experiment. So for a cold room experiment, you have to put the mice. You have to first you have to like make sure the mice are fasted, aka they haven't eaten. Just so you don't have any like food digestion affecting their temperature, like temperatures, because you know when you digest food, you can be a little bit more warm. You can be a little bit warm when you're digesting food. So we want to make sure all the food's out of their system. So you do like 8 hours of fasting, and then we would bring them to the cold room, which is just like a refrigerator. Just think of like a walk-in freezer. We'd bring them there, and you have to leave them there with no food or anything, and just leave them in a cage. And then you have to come there every 5 minutes for about like an hour and 30 minutes and record how their temperature is doing. And this is oh my gosh. And the way to record their temperature for a mouse, you know, like it's pretty hard to like get an accurate temperature reading of a like even us, you know, you can't just like stick a thermometer on your skin and expect an accurate temperature reading. You know, we usually put a thermometer in our mouth, but a mouse, you can't really do that as easily. So what we had to do was you get your little probe thermometer, lube it up, and put it up the butthole. And so you're in this really cold walk-in freezer. There's 16 mice just shivering, and for the next 5 minutes you have to quickly go through all 16 mice, probe their temperature through the butthole, and keep going and going and going, and hope you didn't take too long that it went over to the next temperature collection time. And then you're just there. A lot of times I didn't even get to leave the fridge, because I was like there's too many mice to take the temperature of, and so I'm just like going, okay. back to number one. Okay. It's also so annoying to get the probe. It's so annoying to get the probe up them. I have to do that with some heat stroke people too. Oh yeah, I have heard that. The probe is not so bad to do the first like 30 minutes or so. But when the mice start getting like pretty cold and they're pretty annoyed and I'm cold and I'm annoyed, it starts like going downhill really fast. And by like the last temperature collection, it was incredibly like obvious. Like I was like, I can't like, I don't know, man. Like just put the probe up there, okay? Like please just accept it. For now, it seems like your job was to torture mice. You know, some people probably would argue, yes, unironically. Listen, there was good data to be had. I say that, but I don't think any of my data ended up getting published. I'm pretty sure it's all sitting on my like PI's hard drive, like, because nobody wants to write it, and I don't want to write it. It's been like a few years. It's been a few years already. So, like, in a way, it feels like such a waste that I spent so many years having all this data collected, and it's just sitting there doing absolutely nothing. So the mice really did, you know, they didn't go through this for no reason. They got probed for nothing. I know. The older mice, so because this was like a young versus old mice experiment, the old mice were so unhappy to be probed. They would, I hated the old mice. The old male mice, ooh, they would like try to bite me, and they're like so big too. They're absolute enormous beasts of mice. There would be like, some of them would be like close to 200 grams. Like, it's like fitting the whole palm of my hand. And for a mouse, not a rat, a mouse, it's really, really big. Absolute chonkers of like mice, man. But then like the young male mice weren't any better either, because they just were too hyper, and they wouldn't sit still even when like the temperature gets to them. They're not happy. So I guess since I did cold room fasting is kind of similar to the cold room. I would think about this part. This is I'm neutral, because all you had to do was like take away the food for 8 hours, and then after and this was not so bad. It would be kind of annoying because you have to wake up. You'd have to wake up pretty early in the morning if you want your experiment to be done in a timely fashion. You'd have to like get there either at night to like fast them, or you have to get up like you're doing your lab experiments later in the day, because usually like fasting is not that's it, that's the end of it. It's fasting into data collection. Usually like 8 hours of like no food for them. The only time I would hate doing fasting experiments is when I know there's blood collection involved, because we had to look at like, you know, their glucose, like you have to look at like their blood sugar levels and blah blah blah, and I didn't really like that, because it came into the same conclusion of like, I hate doing blood collection. But it's neutral, it's fine. It's probably because there's not much work on my end. Let's see. Ooh. I'm just like looking to see what else. Lung lavaging. Yeah, blood collection with extra steps. Lung lavaging is like, okay, this one's like pretty specific. I only did this like a few times, and I think I was so bad at it, my my PI at the time was like, yeah, you know, you can just let me know. You can just let me know when you want this done. And I was like, oh, okay, I see. I've been kicked out politely. So lung lavaging is, for the project I was on, we were looking at a specific fungus that grows in human lungs called Pneumocystis carinii. And in order to like study this fungus, we'd have to like, we want to see how the mouse like lung, like what's going on in the mouse lungs. And so usually instead of like sacrificing the mouse and like doing surgery and taking the lungs, which we do eventually, but this is like an intermediate step of like seeing like, okay, little lungs, are you fully infected with like this fungus yet? How are we doing? We would like, you take the mouse, you anesthetize it, can't feel anything, and then you prop it up with this little tool, and then you open up its mouth real good, and then like stick, I don't know what tool you would call it. I can't remember exactly. It was kind of like a pipette, but not really. But you'd stick it right through like right in the trachea, and like you'd give it like a good, like, was it PBS? I think it was PBS. We put like PBS there and then quickly take it out. And then that would be enough to collect like to see what's going on in the lungs. It's really like dangerous and bad, as you can imagine, because you're basically like drowning the mouse. You're putting liquid right into its lungs, and then you have to like quickly take it out, but it can't feel anything because it's all anesthetized. Terribly hard experiment. That was probably the hardest thing I've ever done, like skill wise. Like I think that was just so difficult, and I never really got the grasp of it, and I never got the chance to, because like after 2 times I was told to you gotta go put the fries in the bag. Don't do this anymore. Don't waterboard the mice. Yeah, they were waterboard. Imagine being waterboarded and you can't feel a thing. You can't feel anything, but you're awake. That sounds like hell. I really was torturing mice, huh? I'm sorry, mice. I'm really, really sorry. This data was published, though, so it didn't go in vain, thankfully. I'm not beating the allegations here. I think I would implode if I had to do lung lavaging again, simply because I don't like the idea of like putting a mouse through that when I suck so badly. Maybe if I was like my PI, he did it so like expertly. I would take so long to prepare. And you also have to go to you have to go to the super duper clean rooms for this. Like, so when you go to a mouse room, this is actually go to vivarium. I'll put that next. So mice usually have to be like in a very special housing in universities and research facilities. And so this special housing really requires you to like suit up. You have to be like in the super duper PPE, like the super duper PPE. If you can't tell, like you have to wear like the hairnet, the coat that's over your clothing, gloves. You have to also put like shoe covers on. And this could also like differ depending on like the extent of the clean room. So the university I did this like lung lavaging experiment. You also had to go through like you had to go through like a machine that like a room that like blasted you with air to clean you off. And then if you went to this, like to go to this room, you basically couldn't have gone to any quote unquote dirty rooms. Because you really, as you can imagine, since you're putting like the mice through something pretty harmful, like the lung lavage, especially, I think also this is due to the fact that we were working with skid mice, which are immunodeficient mice. So you really didn't want to like, give them any kind of contamination whatsoever. So yeah, there's like different levels of how clean you have to be to go to the mice. And sometimes you can take the mice out of these clean rooms, and then you can bring them into your labs, and they're considered dirty forever. They can't go back to the vivarium in all cases I knew, because they could have harmful bacteria or pathogens that could be introduced to the other mice living with them. Anyway, I really hated suiting up for the vivarium. Something about having to go to the clean rooms and the vivarium really, like, I don't know, it's not crying in the, like, I guess it was crying in the morning. I don't know, something about, like, having to suit up and go through the horrors of, like, going there. It was always really far away in all my cases, in every like lab that I've been in. Maybe if it was closer, I wouldn't have this much dread. But it was always like a quite a bit of a walk away, and I think I hated it. And I would cry in the morning when it was like I have to go see like the clean room mice, because I knew this meant like I had to do some protocols I didn't like either. I'm trying to like unbias it from like I'm trying to like untangle it from the protocols I don't like. But I also just think I hated being in the vivarium because it's literally walls and walls of mice, walls of mices and like mice in cages, and it smells. Even though like it's like you have to wear a mask and everything, it smells terrible. And I would also be so annoyed when I don't when the when the animal technicians are like they have to like, animal technicians, they're there to like, feed the mice, make sure there's no harm to the mice in everyone's cages. Like they'll send you a report like, "Oh, your mouse is sick," or, "Oh, hey, your mouse gave birth. It needs to do like you need to like separate the babies and all that." I would really get annoyed when I spent all that time like preparing mentally go to the vivarium. I get dressed, I do all the things to get in there, and then I go look and I see that the animal techs are like cleaning the cages of our room that our mice are housed in, and it's not the day that they usually do it, and I'm just like, I did all of this and I have to go back now. And then I just wouldn't go. I literally would not go back that day because I was like, I don't know when they're done. Sometimes it takes hours for them to like finish like, cleaning the mouse room. It's the vivarium stuff is such a time sink too. Oh my gosh, that place is a black hole. I would go to, like, get suited up, do whatever I need to do in there, and I feel like I would enter at 12 p.m. and I come back and it's like 5 p.m. and I'm like, what happened? I thought I was just like, I don't know what, I blacked out in the vivarium, I guess. I don't even know what would cause, like, I don't even know what would happen sometimes. Oh, it's definitely, so I would put this in crying in the morning, and I'm gonna go, where's mouse breeding? This is hell on earth. I absolutely despise managing mouse breeding. Okay, so mouse breeding, it's obvious, like, you have to breed mice. So this is like, think of it as like a silly like game of like, my female mice has these traits and my male mice have these traits, and I gotta figure out which female and male mouse to like ship together in their cage and like produce babies that have the optimal traits, like mix of the traits from one mouse, from the female mouse and the male mouse. And this becomes such a nightmare when there's like so many genetic modifications you are looking at for these mice. So a lot of mice, a lot of times when you're doing mouse studies, there's like genetic components to it. I personally didn't even have to worry about this because the only aspect of that we cared about for my mice in my projects were, are they old? Are they young? Good. That's about it. But I was kind of like forced forced to help with like other people's mice breeding projects, and they had so many like genetic like knockouts, which is like when certain genes that the mice usually express are no longer expressed. And I had to like, I had to help, and I naively thought, oh, it shouldn't be so bad. And it's such a nightmare to keep track of who's doing what. They have to have this certain gene knocked out while also expressing this other gene that's knocked out, and oh my god. And then I'm like, this is definitely like, I will implode if I have to do this again, just at the thought, like, because that made me that like literally made me like throw my hands up in the air. The breeding plus genotyping is terrible. The double knockouts, I know, the double knockouts were horrendous. When I had to start thinking about like Cree, like they needed like the Cree promoters, and oh my god, I'm starting to think you were actually torturing yourself. I think it was torture, and I spent so many years as a bench scientist going like, why don't I like this? Is the problem me? And I think it's just I just this isn't for me. There's some people who eat this up though. So like it was just obviously not a good fit for me. But I knew somebody who like lived and breathed for doing a lot of these experimental protocols. Ah, thank you for the head pat. I felt really bad because I was in charge of so much mouse breeding for a lot of my like lab mates who were too busy to do it, and I really, I think I really messed up their mouse breeding. I remember when I was transitioning out of grad school, I like gave them my breeding like note journal, and I was like, I'm sorry, but I don't. I handed it, and I was like, I'm sorry, I don't think this is right. You might want to start over. And she was just like really confused. She's like, weren't you doing this for like 6 months? And I was like, I'm sorry. I'm sorry. I'm sure she's doing fine. I'm sure she's doing fine now. I guess this goes into genotyping. So genotyping is like the next step after mouse breeding. So you do all this like shipping like calculations of figuring out which female should be with the next male, and then you do all that, you wait for the babies to be born, you take care of the babies, you make sure they're not getting eaten by mom or dad. It's all good. You see the little pups grow, and then you stare at them going, I really hope one of you has the thing I'm looking for, and I hope you guys aren't all one gender or something. And so the time comes when they grow a bit older, and making sure they're not too in. Yeah, that's also another thing. Making sure they're not too inbred. Hello, don't be. And so then you snip. You have to do a little snippy snip of the end of their tails. I did the end of the tails. I know some people like do like the mouth, the ears. I was never a big ear person. You have to snip a little piece of the end of their tails and then dissolve it. And then you have to also mark. You have to figure out how to like you have to figure out how you're going to like identify which mouse is which after you you get the results, which is something I really sucked at. I don't know if anyone else sucked at this, but every time I feel like every method I tried was so bad. I tried ear tagging. The ear tags would just like fall off, and like the mice would just, or they would like chew on their ear, and like they hated it. They hated the ear tags so much. And I didn't know what to do. I just started like marking them with markers, which isn't like the best because it like fades. But I was like, you are mouse number 3 with the 2 black lines on your tail. No one had a good solution for me, so I was just making things up as I go. Anyway, you do all this. I think then you get the tail, you dissolve it. I was thankful we had some funds. We had some funding to be able to send the samples to like a company to genotype them for us, which I really, really enjoyed, because when we had to do it manually, I would really implode. Especially when it's like, it was like sometimes you had like 50 samples you had to like look at, and I was like, oh my gosh, I hate this. But we would send them to a company, though I think my PI who was sending them to a company, he He really didn't. He was trying to like tell me, hey, you know, maybe we should do this manually. It saves a lot of money. I was like, haha, no, we're sending it. It's pretty expensive. I think it's like, I I can't remember the cost off the top of my head, but anyway, as you can imagine, anytime you have a company do something for you, it's always really expensive, and your lab would probably want you to do this by hand rather than ship it off. But no, I said I'm gonna do it, which is it's kind of funny. I said all that. I was like really like I said, no, we're going to do this. I want the company to do it for us, and then I feel like I don't know why I wasted their money, considering my mouse like breeding scheduling was all wrong, I'm pretty sure. And we were never getting the right mice because I was doing something wrong with like pairing up the mice. I just okay, maybe I was just like a bad scientist. I feel like I was just like a black hole of money wasting. Oops. But you know, this is like it's mutual. They don't have me and I'm not suffering anymore. This was neutral simply because I didn't have to do anything. But if I had to do that manually, it's still probably neutral. It's not the worst. I hated doing the gels though, which we can I'm just like as you can see how a lot of these are interconnected. So if you're doing the genotyping manually, oh, we have an ad break coming up. It's a good time for me to like drink water and probably stretch. I'm gonna pause on the tier listing until then. I'm just looking at what we've gone through so far. I see. We're an hour in and we're like halfway through, like less than halfway through. So you know, maybe this was a good length of a good length. I think you know, I like is it gonna be all mice whispering? Not all of them. We're getting through more. We're slowly crawling our way out of mice whispering. I just think I did so many mice related things. I think it's because my last thesis, like my last lab, which was my thesis lab, was heavily mouse based, and most of my like interactions were mice, mice and 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 mice. That like my brain when I think about science is all mice pilled. I feel sometimes I do wonder if I went to a less mouse heavy lab, would I have maybe enjoyed it? But I also think I didn't really. I think even if you like remove the mice from the equation, I probably would have found something. I probably would have realized I don't like doing certain things anyway. Yeah, I'm mouse. I am mouse brained. There was like when I was first thinking of like VTuber concepts, I was thinking at first like a mouse because of how mouse heavy my research usually is. But then I was like, it's kind of passe to do science mouse girl. So I wanted to be bacteria because I do like bacteria. I would kind of wish I was able to join a bacteria thesis lab, but they were so hard to find in my grad lab. Like all of them were all the ones I was interested in that seemed like interesting, at least like at a glance, were so they were full because I think the professors were really popular. And so I'm like, dang, I wish that was me. I wasn't I was not like special enough to be made like an exception. Cause I remember I talked with one professor and I was like, hey, I'd really like to do a rotation with your like lab. I think bacteria are cool. And they said, no, sorry, we're not really doing rotations. But then I remember I saw a girl in my cohort actually do a rotation with her and I was like, they hate me. She hated me. But then I think that girl who got chosen to do a rotation went on, like, she went on to, like, choose that lab as a thesis lab, and then she got, like, a lot of rewards and publications really early on. So, like, maybe that professor just has a really good eye of who's worth picking. Because if she picked me, I might have, I might have dropped out, and that's not good for her. So can I blame her? No. There, professors have an eye for this thing. I did do like one bacterial rotation lab in my grad school time, but I think I just picked something that wasn't really fun. I talked about this in my like science journey stream, but I worked with like tuberculosis for a bit, and I really don't remember. I don't remember anything I did in that lab. I was even trying to remember what I did in that lab for this tier list, and I looked up protocols and papers by that PI to see like what was he working on? And I still don't know. All I remember, like, I looked at the papers and all he was looking at was like OD 600s of like tuberculosis, and I was like, is that all I did? Did I just look up like how much the tuberculosis grew? No wonder I don't know. I don't know. Okay, now that the hand rank is over, I can talk about gel electrophoresis, because that was related to doing manual genotyping. So one thing I didn't like about manual genotyping was setting up the gel, but this is like more related to how many mouse samples I would have. But because when we would do mouse breeding and we would have all these babies pop out, there would be like so many cages I was maintaining, and there was at least like usually like 30 samples, at least 30 samples that had to go through the gel electrophoresis. And gel electrophoresis is basically, let's say we took the mouse tails, we dissolved them so only the DNA is remaining, so we can look at what DNA is like being expressed in these mice. I'm trying to simplify it. And so now we want to separate all the DNA through the gel electrophoresis, which separates things by their weight. And you just like put samples into this little agar gel that you put together. It's not so bad. Putting it together is like the simplest thing. You just microwave some agar and like buffers, heat it up in the microwave, throw some like dangerous carcinogens into your beaker, wait for the gel to form, and then then you put your DNA samples into the little holes, and then it runs an electrical current that separates things out by molecular weight. And it's not so bad. It's not so bad. It's like the most basic baby like biological like technique you can do. I think like most people do it in their biology labs, and it's okay. It's fine. I'm neutral. I would like get super sad when I have to like I had to do manual genotyping, and I would do a gel, and as I'm bringing the gel to get detected, like you have to like, I think it's like usually UV light. You take it to the UV reader, and the gel would just break apart in my hands, and I'm like, because I dropped it or something. And so all your hard work just gets dropped and falls to the ground. But that was rare. And that's usually like that's my fault. I'm clumsy. Not the worst. Would I even say I enjoyed it? I enjoyed it. I think I enjoyed it because I knew that meant it was an easy day. It was like, if this is like all I had to do, like if I had to just make a gel, I mean, yeah, I could do that. I would enjoy this. Well, do I love it? No, not really. But I enjoyed it. But that comes into cleaning. I think that was the one part I didn't like. I just thought about cleaning because, like, I feel like there was usually a mess after doing gel electrophoresis. There's like gunked up agar, like the hardened agar gel somewhere. There's like this annoying contraption full of buffer and a gel, like a, a gel. And so I was like, okay, now I gotta clean this all up. I'm pretty neutral. The first good one, yay! Yeah, with celebration, it's not so bad. I think cleaning was pretty neutral. I think sometimes some things were really annoying to clean. Like, I didn't really like... Some experiments are just prone to being really messy and not great. I'd be a little annoyed at how, like, oh, there's so many parts to clean or put away. It's surprising how much cleaning there is in lab. I feel like sometimes I would just be doing... I would, like, spend, like, 30 minutes, like, after an experiment, just, like, putting things away and cleaning up my bench and cleaning up little pieces and doodads all over the place. And I'd be like, okay, I guess this is my job. It's crazy there's, like, no real good way to automate a lot of that, but, you know, that's fine. I would have put this higher and I enjoyed it. But I think it gets dragged down because of mice. When you work with mice, oh my gosh, everything is so dirty. Ah, it's such a pain. Especially when you bring mice into the lab. When you bring mice into the lab, they're just like peeing and pooping everywhere. When you have to like put them to sleep, they're pooping and peeing all over the cage that they're going to be put to sleep in. And I'm like, this is so gross. Stop peeing everywhere. But I get it. They're scared. Is it just their form of rebelling? I think it's rebelling, and also they're little guys that are grabbed by giant being, and also they're being put to sleep by their own in their like against their will. Oh, also, like, cleaning, like, when you do something like tail blood collection, if you can't tell, you have to kind of restrain them in a way, and so you put them in like a plastic cone, and they just pee and poo all over that plastic. And it was so gross. I saw like one of the cones that we had, and it was like covered in like yellow and brown substances that like would never ever come off, and I'm like, this is nasty. This is the nastiest thing I've ever seen. It's so gross. Mice are just kind of gross. When you go to a lab that works with mice, you can tell immediately. It just smells like mouse. It doesn't matter how well the lab cleans. It's mouse. It's mouse all over it. Mouse there, mouse here. Nasty, nasty. Oh, this is like the first one that's not very mouse related. Autoclave. Okay, you guys like meme about throwing me in the autoclave all the time. Ha ha ha. Throw the bacteria girl into the autoclave. Ha 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 ha. Okay. Know if mice are better or worse than ferrets. I had a pet ferret growing up, and it was pretty bad. I think ferrets are worse. I think mice are just in the setting of a lab, mice are bad because of like the quantity of mice like you're interacting with. And so like usually like let's say you just got like 1 or 2 ferrets, whatever. They're pooping and like they're pretty nasty. The one ferret I remember, I don't remember it too much because I was pretty young when we had the ferret. But it was like he was pretty stinky. It's pretty reminiscent, I guess, of a lab mouse stench. But I think the lab mouse stench is only that way because there's like hundreds of mice coming in and out, pooping and peeing. They come in and go, hello, it's time for me to pee and poop here. Thank you. Autoclave, yeah. So the autoclave, you guys throw me in here. It's to sterilize. You use this to sterilize and heat. Sorry, there was like a loud plane and it distracted me. They so autoclaves, I don't really like using them. I don't know if anyone else feels similarly, but when I would have to prepare things for autoclave, I really hated using the machine. It was really gross. It was really gross and also really loud, and it was really our autoclave, nothing like this picture. It's like a giant like industrial autoclave. Like the room was dedicated just to the autoclave. It was always full. Like it was hard to schedule like you had to schedule a time, and everyone always wanted to use it, and no one would wait for the autoclave to be completely full, and so people would just throw in their one little dish tray, and I'm like, buddy, I need to also throw a little dish tray in there. I just hated I hated handling autoclaving like things. This was like such a like I would push this to other people. I'd be like, hey, can someone else do this? It's just so annoying. This is maybe I needed a tier between crying in the morning. Okay, I'll just put crying in the morning. Are the lab mice different than wild mice? I know the US lab ferrets are super different than the wild EU ones. Yeah, they're pretty different. I think, like, lab mice, like, versus a wild mice, they just have... I think they're considered... I don't know, like, off the top of my head. I know there's, like, biological characteristics that are different, obviously, like from a wild versus lab mice, just because these mice have been like taken care of in basically a sterile environment, and that their microbiomes are different. I think they're like characteristically they are more calm. I actually don't know. The inefficacy of the autoclave process sounds annoying. Yeah, it's just like, I think if we had an autoclave that was, like, if you can see in the picture, it's so teeny tiny. If it was just for me to use in my lab, I was like, oh, that's great. I don't mind doing that. But to, like, have to, like, share it with the whole floor, and people are, like, really ineffective, or they don't follow the schedule, it's just so frustrating, because you just have to sometimes wait, and I'm like, oh my god, let me in, let me in. I didn't really have to autoclave too many things, at least in my, like, thesis project lab. I don't, like, this is, like, a lot more common if you're doing certain, like, techniques and protocols. I think this is a lot more common with, like, especially bacterial labs, because, you know, you're pouring plates a lot of the time. The most I would usually use the autoclave for was to sterilize my surgery equipment. So a lot, like, you know, you can't use dirty scissors and scalpels on like mice. But it was really rare that I even had to autoclave it, because we just had so many bags of sterilized like equipment that I would just like go through before I go like, okay, I guess I've run out. Let me open up a new pack, or let me autoclave a new pack. This could go into I want to talk about tissue extraction. Yeah, so tissue extraction this is basically the mouse is no longer with us, and we must now harvest the tissue. And this involves, you know, you have to basically open up the mouse, and I was... Good night, Phil. It's past 1 a.m. Yeah, good night, good night. I hope you have a good sleep. Bye, 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 bye. Tissue extraction is not so bad compared to, like, surgery. Because, I mean, the mouse is unfortunately gone. But you can kind of be messy-ish. You know, like, when you're doing surgery, you have to be very mindful, especially if it's surgery where the mouse has to survive. Have fun reminiscing about your rat whisperer days. Yeah, thank you for hanging around. I appreciate you. Tissue extraction, like, it's just like you open up the mouse and you collect whatever you need. In my case, we usually were collecting lungs and adipose tissue and spleens. Spleens are usually like a super popular one. If you are a mouse person, you probably collected spleens because splenocytes, which are spleen cells, they're like chock full of like usual. Usually, they're controls for a lot of experiments. Like when you're testing like what's going on in one tissue, you'll take like the spleen and go, okay, and this is our control tissue. It's I hated the spleen. I think it's I was primarily an adipose tissue person, and adipose tissue is a lot easier to like collect and extract and purify and isolate the cells from compared to like the organs like spleens. So I would like adipose tissue is also kind of funny. It's kind of funny to collect. So we would take the subcutaneous fat. I would also take as much brown fat, which is like the fat that's found like in the clavicle region of the mice. I would collect that. It's a pretty small amount depending on like the age of the mouse. Little tiny little bits of like fat. But the subcutaneous fat and visceral fat, that was like huge. It was like you could collect like a giant chunk of it when you open up like a fat mouse. Ever study anything with their brain? No. I've actually I don't even know like how you would go about like getting to the brain. It's kind of interesting. I don't know how many kind of gross to say. I opened up quite a lot of mice for tissue extraction, and I don't think like I would know how to get there, like how to get to the brain without like ruining the tissue. Which is, I guess it's such a highly specific skill. The neuroscience people truly are doing their own. They're living a different life than other people. I'll have you know. I liked tissue extraction. I don't know if that makes me a bit like crazy, but I think it's just because it was really simple and straightforward, and there's not much to do, not much else to it. It was just literally like, oh, okay, I open up this mouse, I get it, and then I put it away. The only like downside was it when I had to like do spleen collection, but even then, it's not the end of the world. That's the only part another streamer I watched analyze had to do with antidepressants or something. Oh, that would make sense, like why you would open up the brain there. I've always like the brain, like neuroscience and brain work is like super interesting. It was really hard to get into that like department though. And I don't think like unless you were in the neuroscience department at my grad school, I don't think you weren't allowed to do like any rotation in the like anything related to brains because there was just so few like researchers in our school that were even doing neuroscience lab work. So obviously the neuroscience students should have priority access to that, which makes sense. And I think it was like if I remember correctly, like my school had an umbrella program for grad school, which basically means like, anything under like this umbrella of biology will go like, you are in this department. But then the neuroscience people were the only ones like that were outside that umbrella. They were so special and you had to do like special requirements. It was crazy. Could not be me. Could not be me for sure. Bacteria culture. I really, really love bacteria culture. I know that's not surprising as someone that's a bacteria, but this was just always so like relaxing to me. There's like no stress to it. This is probably because my work with bacteria was highly, highly limited and very rudimentary compared to the, I think, extent I went to with mouse work. So I think this is me looking at things with a rose tinted glasses, so to speak. But I loved working with bacteria. It was just so relaxing. There's not much thought. You just like no one's biting you. You just put things into the incubator. You put the thing in the incubator, wait for it to grow, collect it, and then extract and isolate. Ah, it's so easy. There's no like there's nothing else to it. Okay. I know there's gonna be like a microbiologist who actually does bacteria, like who did extensive bacteria work, going like actually. But when my experiences with it were very straightforward, and I would take that any day again over doing mouse work. No one's biting me, and no one's peeing and pooing on me. Okay, and this is also less like mouse related, but it's kind of mouse related. This is like more cell separation is more of the downstream protocols after tissue extraction. So like after you get the tissue and now you've isolated the tissue cells that you're looking for. You got rid of all the gunk and doodads. Now you need to separate for the specific cells you're looking for. And usually in my case, this was like specific immune cells. And so cell separation, there's like columns and usually magnetic beads. I didn't, this is like, this picture is specifically the magnetic bead separation, the max one. I think there's other forms of cell separation, but this was like my only experience. This was my only experience. And I I really hated this. It's kind of hard to say though, cuz like I think it was I think that there's a bit of bias about how I don't like this, simply because of the fact this usually was like one of the last steps of the day for experiment wise. So like to lay out the idea of like what a timeline of like what a day of experiments was. Hold on. I'm gonna do it. So I can put it back. So it would be like fasting. I would do fasting into cold room, into tissue extraction, or into tail blood collection, into cell separation. And so I did like all of these things, and so cell separation would be at the end, and I go, oh my gosh, I hate this. I think I messed up. Delete row. It's definitely not. Yeah, I think I would just be like really tired and want to go home. And since, like I said, it's like a column that you put all your samples in, and like it's slowly like dripping. It's dripping and dripping and dripping, and it's separating all the junk you don't care for. But I'm just sitting there looking at it, going like, "Can you drip faster? Can you hurry? Hello, please hurry here. I just want to go home so badly." And there's something else. You're just usually cleaning while waiting, and it's not great. It was just so like it's just so boring. And oh my gosh, I think this is also because I'm tired and it's the end of the day and I'm wanting to go home. I was prone to making mistakes at this step very often. And this is a stage where you really don't want to make mistakes. Because this is where all your samples are. This is what you did. This is all the things you did led up to this moment right now. And I think there was at least 2 instances where I spilled spilled something, or like put something in the wrong tube, and like everything I did it's just gone. It's just done. There's no going back. There's nothing to do anymore. I was just like, and then then it's like, well, I guess I'll restart this experiment next week. That's obviously like a skill issue. Nothing to do with the experiment itself. I think a lot of this is, again, these are all my personal experiences and how I was feeling during each of them, like while I was a scientist. And it's hard to like untangle the biases of my personal experiences from this. Facts. Okay, if you don't know what facts is, it's like it's pretty much like flow cytometry, but this is fluorescence activated cell sorting. And if you've never heard of that, it's similar to like you. This is like the kind of like the end of it all. If you successfully separated your cells and then you have everything ready, nothing went wrong, you didn't spill anything, you did it. The next day, usually, this was like a next day experiment, you would treat your samples with certain antibodies that have specific like fluorescent biomarkers. They like go, wee, I'm this color, or I'm this color. If if it successfully finds whatever you're looking for, you would treat your samples with that, and then you take it to this big flow cytometry machine, which you had to schedule for in advance, and then it will read each sample one by one and see which fluorescence activated, like which fluorescence it's giving off based on, uh, you know, what markers are available. So, if it has XYZ marker, then it will fluoresce with XYZ colors. And this is honestly, it's a really cool experimental protocol. It's very, very like quantitative, like because you're looking at it at a one cell at a time, and you're seeing which proteins or whatever you're looking for are being expressed. You get a lot of really good data from this, but it is really complicated. I think you can get a job just being like just doing this. Like you can just be like the director of like flow cytometry and help people set up their experiments with this because of how complicated it is. There is so much planning to go on this. So like I said, there's like fluorescence that activates with each biomarker present. And you have to like carefully plan which fluorescence you're looking at, like which fluorescence you're going to use for like which biomarker, because as you imagine, like light and color is a spectrum, and you want to make sure, let's say, certain colors aren't interacting with other colors, so you don't have like a mixed signal. And so if one color is too close to another color, you wouldn't be able to really tell very easily which biomarker is being expressed, and that would be a problem. It's a really, really cool experiment. It gets complicated as you could think. Like, let's say you start saying you need to examine like 12 different things being expressed, and now you have 12 different colors you have to be mindful of, and you have to think about like, well, you have to think about biologically, like, well, where is this being expressed? Is this protein expressed closely with another protein? So should I make sure they're not similar colors, or should they be really different colors? Or is it okay if they're similar colors because you wouldn't normally find them together anyway? It's really, really complicated. It's really, really interesting though. I'm gonna actually put this in love it, thank you, because I just thought it was like, I think it was one of the most rewarding parts of doing all these experiments. I would go through hell doing all these things I don't really like, and I'd be really happy when I get to the last part, which was like the facts sorting. And the analysis was cool too. So you can see like it's these like heat, kind of like heat colored plots here, and so you would like have to do different ways of understanding, like, okay, I'm looking for expression from this and this, and we're looking at the intersection of when those two things are expressed versus, oh, I want to make sure I don't see any Z expression, so let me make sure I'm only looking over here. It's really, really cool, really hard, really difficult. I think you can spend so many years like working on that. And I think that's why I enjoyed it. I like I had to get a lot of help to be at like to understand that part of my experimental process. And I like really that was like so enjoyable. I think working with the directors and like core for that experiment for the flow cytometry was always super fun. They were so knowledgeable, and it's crazy how it's crazy how passionate they were about flow cytometry. I loved it. Would do it again. Would just join would if I like I think if I were committed to doing like a PhD of some kind, it would just be so I could go work at one of those cores, because I think you have to have a PhD to do that. Or maybe not, but I imagine you probably need to be very, very, very experienced. Very, very experienced. Definitely not anywhere close to be experienced enough to like apply for one now. But maybe if I'm bored. Okay, I feel like I just went talked about something really complicated, and now we're going to pipetting. Pipetting is I don't know why I even put this here, but you know, pipetting is like the nature of like literally all these experiments. You just pipette. You have these little special pipette instruments, and they can give you dispense certain amounts of volume down to the microliter. Really cool things. I kind of want one. I really want to buy like a scientific pipette, a micro pipette whenever if I could find a good deal for one. They would not be very accurate though, but it would be fun for home use. Also, I just want to like pose with it because I think it'd be cute. I think I was not very good at pipetting, which is why I tended to have problems with molecular biology experiments where you needed to pipette very accurately. And I would complain like, I think my pipettes are bad. They're not calibrated. But it was not a pipette issue. I think it was I'm pretty sure it was a skill. It was I was being skill gapped. Pipetting is like just the basis of it all. It would there is one thing I would hate is like some days you're pipetting so many things and you're doing it just so often. You would like start to hurt my wrist and my arm because you're just like doing these like same movement over and over and over and over. It was like a recipe for like, you know, hurting like RSI and all that. I was also I remember I would people did not like how I took pipette tips out. Cause so when you I hear a lot of people really love their fancy pipettes. That's true. I think the fancy pipettes, they're very, very special. You want to be like, I think a lot of people are very protective of which lab pipettes they own too. I know personally, I was like, these are my lab pipettes. Don't touch them. Because they're easy for them to get like damaged, and you don't want to like unknowingly have them damaged because someone happened to borrow one, and then you do an experiment, and it's like, oh my gosh, why is everything so off? And it's because someone like broke it and you didn't know. Like, oh, I think everyone in our lab like had labels on their pipettes, and it was like, oh, don't use the pipette that's not yours. Pretty it is pretty silly to think about it like that. I like mugs in an office. Yeah, like mugs in an office, but definitely more. I guess it feels a little sillier because it's like if you can't. I'm sure like some people, no one's gonna really break your pipette, and it's not like it's that personal. But I get it. Nobody. You want to know what's happening to your stuff. It's your precious thing. Analysis. I hated this. I I I don't this is funny because you would think like as a science writer now who does mostly just like reading papers and looking at data, analysis shouldn't be that painful. But I really, really hated date like when I finished like collecting all the data that I needed, it was time to just analyze all, like, everything going on. I was like, oh my god, I hate statistics. I hate making graphs. I hate putting stuff together. I don't think I really ever understood, like, stats. No one... Everyone tells me, like, their PI... Everyone would tell me that their PI, like, would help them. I don't think I ever experienced, like, a PI of mine, like, sitting down and explaining how to do statistical analysis for, like, my lab experiments. I was always told, like, it's time for you to figure it out. I'm like, I don't know what's going on. That is someone's full-time job. Yeah, I know there's some labs, they just send their data. They send their data to some guy, and they'll do all the analysis. I wish we had that. I've never experienced that before in my life. There's, like... I remember I, like... this is a painful memory. This sounds like a painful... It's a painful memory, but I'm laughing at it because I think it's kind of silly in retrospect. I remember I put together an Excel sheet of, like... I think it was my qPCR data analysis. And I was like, I've never done this before. And I, like, really worked hard on this to, like, understand what this is. And, okay, and then I presented it to my PI, and he's like, this is completely wrong. I don't understand how you did this. This is terrible. And I was just like, ah. I didn't know how to respond. I think we ended the meeting right there. He didn't even want to explain it to me. He just told me to go do it again. They just sound lazy, not wanting to put in the work needed. Yeah, I do think it's just, like, a matter of... I don't think they were that good at being teachers. Which is, like, the unfortunate case for a lot of these research professors. They're not really teachers. They're good researchers. That's why they're here in the first place. But teaching is not their forte. Which sucks. I don't think I ever even got that PCR right. I won't go into it. I won't go into it too much. But from, like... I don't think that PCR analysis never was completed, and I think that was, like, a point that led up to me going, like, yeah, I need to get out of here. I need to get out of here. It was even, like, he asked me, like, can you give me your Excel sheets so I can see what math you did? Because it doesn't make sense how you got here. And I'm like, you don't trust my math? I mean, I think the math is wrong, but it's not like I made something up. Well, hello? Drug dilutions. Drug dilutions, like I said in the beginning of this tier list, I think this is more like... I should say this is, like, lab math in general. I really sucked at drug dilutions. There's, like... I feel like I just never really... I know it's C1V1 equals C2V2, which is concentration of your original, and your volume of your original equals the concentration of your your desired concentration and your desired volume. I know that, but when I had to, like, consider how much... You have to consider also in, like, a physiological sense how much of the thing you're putting into the mouth, because that was usually my case. Like, we're injecting this drug into the mouth, and you have to be mindful of the amount of volume, and then the interactions, and also how you're going to give it, like, how are you going to, like, dilute it. Something about it never, like, really stuck with me, and I was just like... I figured it out once in a way that worked, and then I went, like, from there. I just kept copying the same number, because I was like, well, that worked, and I'm never gonna change that again, because I need to... This is the same experiment over and over anyway. I just sucked at lab math, I think. Something just didn't stick. And all my PIs didn't want to teach me anything math related, so it's like, whatever. I'm not asking for any help. I'm just gonna figure it out once and never understand why it worked like that. Lab math, too hard. Math is just too hard. I wasn't here to be a mathematician. I was here to be a scientist, but then I didn't even want to be a bench scientist. So from drug delusions, usually it was time to do an IP injection. I can talk about IP and IP injections. The IP injections are intraperitoneal injections. So this is like tummy. Think tummy, but not really. You don't want to inject right into the tummy. You're like injecting into the space between the subcutaneous tissue and the tummy. Just like the empty space. This is like the easiest. So you would just like have to restrain the mouse and scruff it by its like neck and then give it a little lift. Hold the mouse back with your like pinky to make sure it's not like the tail isn't whooshing all over your hand, and then you give like a little injection onto its tummy. This was like the easiest. I think the hand technique is a little tricky. I knew like when I had to teach people how to hold the mouse for an IP injection, I know some people like they really struggled with that like hand holding. You really have to kind of like restrain the mouse with your free fingers while also scruffing the mouse's neck so it can't actually like so it's not doing this. Like when you restrain it properly, and that's like it's like it just looks at you like that. This was like really easy. I loved doing these because usually it meant when I had to do IP injections, that usually meant there's nothing else to do because we're just waiting for the therapeutic to like have its effect. Enjoyed it, did not dread it, and I meant okay, that's it for today. I just like I make the drug and then I inject it and then I'm off. That's it. Bye. Actually, I couldn't actually leave because no lab PI is happy to see you leave before 5 p.m. But you know, in my mind, I was free. And then the opposite of that, it's not really like an opposite, but like opposite in like how I was feeling was IV injections. So this is intravenous injections, and so instead of the empty space in its tummy, which is like very generous and like it's very generous, it's really really generous. Now I have to like get to the vein of the mouse, and you know, if you've ever done blood collection for like a human, it's hard to like you have to kind of like restrain the arm and find a vein. It's pretty difficult. You can't really do that with a mouse arm. You have to restrain the mouse in like a little plastic cone, similar to the blood collection, and then you have to find the veins on the tail, and you have to be careful of certain veins, because certain veins, if you inject into it, they will just die. I've experienced that. I didn't know that until like I did it, and they just died. So you have to hold the tail and find the veins right on the mouse tail, and it's so difficult because like if you feel any kind of any kind of resistance, that means you're not there. And so I just had to keep like re-pricking and pricking and re-pricking and pricking. And I felt so bad. Very small veins. They're very very tiny. Mouse tail veins are like super tiny. They're very visible though. Like if you give it a little rub, you can find like the blue veins, but they are really tiny because you can easily overshoot, and then now you're just in the tail, and that's nothing's working if you're just in the tail. You have to get into the vein, and you can't keep re-pricking them over and over. So after a while, if you keep messing up, you have to go, okay, take a rest. Because if you don't let them like rest, they're really scared in their little plastic cone as you keep pricking their tail over and over. And sometimes they just die because they're like so scared and shocked, and like I've had that happen unfortunately as I was learning how to do mouse IV injections. They just like I would go like, "Woo, I finally did it." And then I noticed that the mouse is not moving in the cone anymore, and they just passed away because they did not happen. They were so scared, and it's so sad. It's not only like my experiments like frick now because I just lost a mouse, but I would just what a terrible way to die. They were just like so scared in this little tiny container as some large unknown being is like pricking their tail over and over. Awful. That's definitely like I'll implode if I had to do that again. I could not. I could not. It was really difficult. Oh, kid experiments. Okay, this isn't like much of a. This is such a general one. I think I had a. I laughed to myself when I even put this on here. So, kid experiments are. These could be. There's so many different protocols that use kits. So these are just like companies make a little box full of all the things you could ever possibly need, like what buffers you need and what columns they need, and you follow the directions, and there you go. You got a whole experiment going. I love doing kit experiments. They were just so simple and straightforward. Everything I need is in one box. The protocol is written out for me in the most like dumbified way. I enjoyed this quite a bit. Mice seem so resilient and fragile at the same time. Yeah. It's very insane. Like, you have to be so careful with them, and they can also smell your fear. So you have to be like, you have to be like really like not scared of them while grabbing them, but you also have to be gentle, and you have to not like hurt them in any way. It takes like a very special touch. I can see why when I first started learning how to work with mice, the person that helped me, the lab manager, she said she'd been working with mice for like over like 30 years. And she's like, and she mentioned that she only started feeling comfortable and like calling herself like an expert at handling mice in like recent years. And I was like, wow, it really takes a long time to feel like you can say you are an expert mice handler in the lab. So yeah, in regards to kit experiments, the only reason why I would say this is not love it, thank you, is because of how annoying it is when the buffers run out. They run out like at different times. And so now you have like a box full of buffer 1 only, and then you have to buy a new box, and then you buy box 2, and now you don't have any more of like you don't even have more of this buffer now, and now it's just you just end up with boxes and boxes of these kits. I don't know if other people who have worked in labs experience this, but every lab I've worked in with box kits, like experiments, their shelves are just full of these expired old box kits full of buffers, like little bits of buffers from like 2014 or something like that, because they never run out at the same time, and you just keep having to buy new ones, and I don't think you can buy bottles individually. Or maybe you can, and everyone's just buying the boxes because they can. I don't know, but every lab I've been in has just like a shelf dedicated to 2014 boxes. Oh, okay, now this is like not even a lab experiment. This is just lab presentations. I love lab presentations, love doing lab presentations. I loved talking about my lab work. Caveat, I'm untangling this from the biases in my brain saying lab presentations were living nightmares because people were mean to me. I say people, but I mean more like my PI was mean to me. I just really like putting together a nice presentation and talking about what I was accomplishing and getting like questions from people in the lab about like what I was doing, and also like their input about how I could potentially tackle certain problems. I love that. That was always super fun to me. The caveat, and I'm not going to let this detract from my scoring of it, mind you. It's still love it, thank you tier. Caveat is, when I used to present in my thesis lab, my PI, he would like not say a single thing until the end of the presentation, and then he would make this kind of disgruntled noise and go, okay. And then he just starts firing off complaints. And I'm like, buddy, you meet with me like on a weekly basis. You could have told me these things before I do the monthly lab presentation meeting. Anyway, again, untangling that from this love lab presentations. Not a surprise I like doing streaming, because it's basically like it's like lab presentations over and over again. but way more casual. But I really like. I always ended up making lab presentations way more like casual and friendly. I think that's just in my nature to make things. It's just in my nature to make it not as serious. It's hard. It's hard. Maybe some people didn't appreciate that and thought I wasn't taking it seriously, but it's just like I like it. Sounds fun. Yeah, it's fun. I think though, I think some people find it really stressful, and I can understand that. I only present it like maybe once a month. I know some people, their labs make them present weekly, and maybe that could be a bit stressful if they have to present like new data every week because it's like a lot of pressure to accomplish something. But I presented a lot of like negative data, though, maybe that's not like something that would fly. Depends on the lab, I guess. Prep for experiments. Again, we're going into like way more general things. I enjoyed this. This was like a fun thing. And I'll mention why it's not love it. So when you prep for an experiment, this is something I learned only like in the last few years of my lab bench scientist years, was, before you do an experiment, it's good to do the labels. It's good to label things and like label your tubes and label everything before you actually do the experiment. That way you're not labeling as you go, because, labeling as you go. Oh, hi, hey, Ink. Yeah, you should never like really label as you go. When I was like an undergrad, I would do that, and it was really bad because I was like, you can't. It would waste so much time. So, in like my last few years of like a bench scientist, I like would spend like the last hour of like my day. I would like go through and label literally every single tube, label every single like media, label everything going forward. And the only reason I would say this isn't love it thank you is because it would make me very aware about how much plastic waste science builds. Like I would like have like three styrofoam like it's hard to picture if you've never seen this, but like the three styrofoam cases of like conical tubes would be like labeled in front of me, and I'd be like, wow, that is a lot of plastic waste. It made me too aware of how much waste is being generated. I was like, I prefer like being unaware. Label your tubes, you will forget. Then you have unlabeled. Yeah, exactly. Exactly. You will have unlabeled tubes. It always ends up like that. Someone who does genotyping for a living, you do genotyping for a living, oh, that could, like, the tissue extraction is held depending on your method. Yeah, it definitely depends on, like, what your tissue was. For me, it was, like, adipose tissue, and adipose tissue is really easy to extract. So yeah, I really like the prep for experiments. Again, we're getting into more. Okay, we're finally in a sciencey, another sciencey one. Protein purification. I hate it. I really don't like protein purification. I think protein purification is like, it's so much waiting. It's so much waiting and waiting and waiting. I like would. It was just boring. I didn't like it at all. I didn't really do much protein purification in the first place because this was only like one of the labs I was in was that even did this. But when the lab that I was in that did it, we did protein, like we'd purify like a lot, and I'm talking a lot of of samples, and we'd have this giant like quart size cup. It's hard to like imagine. It was like a giant quart size cup, and we're like waiting for it to like slowly empty out into this tiny like column, and it's like dripping one drop at a time, and I just sit there waiting and waiting and waiting. I was sitting there waiting because we couldn't do it actually overnight because it was you know, I had to do other things immediately after it. Yeah, it was like it's a gross, ew, gross. He used to, yeah, it's just like I can't say this is enjoyable. It won't make me implode if I had to do this again because it's not like it didn't give me like pain. It was just boring. It reminded me of protein postdoc who used to purify proteins out of E. coli. He had this huge setup and it would still take forever. It just like it can take so long sometimes, and it's crazy. Oh, this goes into the next one, qPCR. Okay. I remember I was talking about like I'm trying to untangle some personal biases when it comes to qPCR, like to some of these things. The qPCR, I can't untangle the personal biases. I hate qPCR so much. It's so finicky. It's so like time consuming. It makes my brain hurt because you have to be super careful as you're going through all of these different tubes and like wells. It makes my head hurt. I hate analysis of qPCR data. I hate the machine. And the machine sometimes was just it just broke so often. Uh Again, I also think qPCR is slightly less painful depending on the machine, and yes, in my workplace we hate qPCR analysis too. It's so bad. I think I just like especially despise it when I think about how I think I don't remember which one we had. I think we had the rotor gene. I can't remember. Either way, I hated it. I think I also hate because my PI would talk about how qPCR is the easiest thing anyone can do. It's the easiest thing anyone can do, and then he'd get upset when like my data is wrong, and I'm like, I don't know what to tell you, buddy. I don't know what to tell you. It's wrong. I don't know. I think the qPCR arc was like really the part where I was like, I can't do this anymore, guys. I can't do this anymore. And yeah, that was like leading up to the I'm dropping out. Lung function study. This is like super specific to like the fungus lab that I worked in. So we had to like, we would figure out when it's time to sacrifice the mice and look at their lung, I'm trying to think of the word. We would figure out when we need to sacrifice the mice and like look at their lung tissue expression after by doing this lung function study. So we would put the mice in this little contraption, this little teeny tiny contraption, and we'd connect it to their little mouth, and then we'd have to like we just measure basically how much they're breathing, how much is their lung expanding, etc., etc. This was actually kind of fun to do. I think it's because we were working with skid mice, which are the immunodeficient mice, and they were so chill. They had no energy to do anything anyway because their lungs were full of fungus, and so they were just like. So like when they started breathing really heavily, that's like when we go, okay, yeah, it's time. Their lungs are full of fungus. It's time to analyze the lungs. I'm putting in this as I'm neutral because it would take a while. It's like getting the mice set up to do it. It's not takes a bit. Oh, thanks, Caledonia. Yeah, it was always sad working with the skid mice. Hi. The skid mice were like they're like the white. I feel like they're like the classic lab mouse that I think your average person might think of if they've never worked with science. They are like the white mice with red eyes. Like they're black sixes are the worst to work with. Yeah, I worked with both skid and black six mice, and they were again for me. It depended on how old they were. When they're younger, they're definitely like bitey and jumpy. When they're middle age, I think they're a little bit more calm. But when they get to like the super old age, they're back to being like they're back to being jerks. They're like they're so jerky when they're old. It's so annoying. They just like they can't they're just angry because they're gonna die soon. They just want to bite you all the time. But maybe it's just also men. It's the male mice. The female mice usually weren't that much of a problem, but we had to, we had to be mindful of the gender equality. Not. Oh, this is a really cool one. This is similar to facts. It's up here with me. It's up here to me with like facts. Single cell sequencing. So you know how we talked about there's single cell sorting? I don't really. I'll be honest. This is like a procedure you ask you hire someone to do the actual procedure for you. So maybe that's why I like it, because there was like barely any work we did. You have to like tell them what you're looking for. But I really liked the analysis of single cell sequencing. You get like a Tisney's plot, and it shows you so much information about how what genes are being expressed. It's like the amount of data you get is kind of insane. And I know I said I don't like analysis, but it's like mostly I think I'm just like enamored by single cell sequencing because of like the depth of data you get. Even though I think I was never really that good at analysis of it. Yeah, I can't say I was an expert at doing single cell sequencing analysis, and I think I might have even messed up. I think I might have even messed up a bit, like misinterpreting what data I actually saw in my samples. But I really liked it. I wish this was something I could work on now, just because I think the data's cool. I don't see it a lot though, like in my line of work with cancer at the moment. But yeah, I loved it. Tissue freezing. I put this one specifically because I remember the amount of times I hated doing this. Like, so after you extract tissue, and let's say you're not going to work on that tissue now, you have to go work. You're gonna work on it later. I put tissue freezing here specifically because I hated getting prepared for this. It's something about it like really like bothered me. I think it's because I hated getting liquid nitrogen. So when we had a liquid nitrogen tank that was shared like across the floor, and to freeze the tissue, you need like to get a bucket of liquid nitrogen to throw the tissue samples in to freeze them. You want to snap freeze them so you don't lose any like activity, or like the things aren't dying. You want to minimize cells dying ASAP. You don't want them to die. No dying. And so you do that. Nothing to do with the actual thing. I just hated getting liquid nitrogen because nobody would ever report that the liquid nitrogen is empty. The dispenser was like always close to being empty when it was my time to do it, and nobody would ever fill it up, and I would just have to go, oh my gosh, there's no liquid nitrogen. What am I gonna do with my samples? And then I have to go around floor to floor going, hello, can I please borrow your liquid nitrogen? I hated the sound the dispenser tank makes when you open. I hate the sound too. I just don't like anything to do with liquid nitrogen. I think that's the moral of the story. I should have just put liquid nitrogen. I'm gonna implode if I ever have to work with like liquid nitrogen and tissue freezing again, simply because I know you can't trust people to keep track of the resources, and nobody ever wants to fill things up. Nobody ever wants to report that something's empty. Liquid nitrogen ice cream is amazing. I do love liquid nitrogen ice cream. When I was an undergrad, I was always part of what would I call this? We would have like a festival to show science to the public, and I was always part of the stand that helped make liquid nitrogen ice cream for the people. And the kids were always amazed. They were just like, wow, ice cream. Wow. It was always really, really fun. And I got to eat so much ice cream until like I got lactose intolerance, and then I couldn't eat the ice cream anymore. Gavage. Gavage is similar to lung lavage. I don't think there's much to it. I don't know why. I think I meant to delete this one because they're basically the same. One is like getting the liquid out. This one's just literally putting the liquid in. I hated it. Would not do it again. I already expressed that I sucked at like putting like doing anything intertracheally, getting them like opening the mouth up. It never worked for me. They never. My PI would go like, okay, you have to like you have to open up their mouth like this, and like they stick their tongues out, and then you can put in like whatever solution you want. And I'm like, they're not sticking their tongue out. I don't know how you're doing this. This makes me happy. I was never trained for the animal room. There's some animals were definitely not for me, but I know for some people they lived and breathed the animal room. They loved it so much. I could not relate. I could not relate at all. I don't know what they were thinking. Our animal facility at my own was a shit show too. Yeah, I feel like most people's animal facility experiences are bad. I've been to multiple research facilities and schools now, and all of them seem like they have their own problems. And I think that's because low key, like, nobody actually wants to work with animals. People say they want to work with animals, but nobody actually wants to work with animals. We actually got audited by the ethics committee because the techs didn't monitor the mice enough over a long weekend. 10 drop dead. Oh my gosh, no, not the mice. I remember there was like a scandal at one of my schools with like the animal technicians not doing a really thorough job, and they missed apparently a lot of cages. And I was like, ooh, that's bad. Tissue lysis. Tissue lysis kind of comes like right after the tissue extraction phase. And again, this is really dependent on which tissue you're working on. I'm gonna put this in neutral. Because of the fact I hated certain tissue lysis protocols. Adipose tissue lysis, wonderful. It was really straightforward because I think anything that has blood, really annoying, really painful. Adipose tissue, beautiful. All you have to do with to extract the cells and break down adipose tissue is kind of like centrifuge it a bunch of times. All you do is like put buffer and centrifuge it over and over, and eventually all the fat part goes away. And then you have whatever the cells you're looking for. Usually it's called the stromal vascular fraction. That's when, that's the good stuff. That's where all the important information is. You don't really care about like lipids and all that. Unless you're a lipid biologist. Maybe you do, then you do care about lipids, but not me. But when you have to work with things with like spleens, the spleens take so much. You have to get like a little mesh. You have like a little mesh sieve, and you have to start mushing you have to mush the spleen through the mesh sieve, and then like collect the stuff coming out of the sieve, and then put that in a specific media buffer, and then you work through that. It's like such a pain. And it also worked fast. I think the one thing I liked about adipose tissue was like you didn't have to work that fast. It was pretty generous. In fact, like you even have to heat it up at one point to really get the gunk out of there. But spleens you have to like, it's so like I'll die. I'm gonna die any moment now. That's what the cells are telling you every time. Yeah, so because of like the good of adipose tissue and the bad of spleens. I'm gonna put this in I'm neutral. We're almost making it through. It's only 2 hours. Most of these should be faster. We have 1 minute of ads, so I'll just take a little break. Okay, I think my will implode section is about as long as I expected. But there's more things that I am neutral and love it. Thank you. I'm neutral and above. There's more than I expected, so I'm happy about that. See, I don't hate science completely. Science is kind of cool sometimes. Sometimes it's kind of interesting. Some of the bench work isn't so bad. I miss actually doing research. Yeah, yeah. Are you like technically a technician? I think it can be so demoralizing when all you do is like pipette, and some people I think are into that. I have a friend who's like recently started doing that kind of work, and she's like, I'm all for this. She's like, I love turning my brain off, and all I do is pipette and like mix together chemicals. I'm like, oh, cool. Yep, edit genotyping, and that is all we do. Yeah, it's kind of. I'm sure you'll get a chance to get to do real research eventually. It is like, I think there's like charm to like turning off your brain. But then I do agree, like I would miss intellectual like stimulation. Honestly, the upside is I have a ton of freedom. The downside is I can already feel the cobwebs forming in my brain. Yeah, like it feels like sometimes, like, especially if your goal is to be like a researcher of some kind, like time away from doing like actual research is so hard. I think I took a bit of a break in grad school. I had a little mental health break for like about a year or so, and then I went to what became my thesis lab. And it was so rough just in that 1 year of not doing anything, and it was like I feel like I had to relearn everything. But obviously I was not even in a lab, so it's a little different. Okay, this is not even a lab technique or anything, but I just want to say I love the vortex. I love spinny thing. I love little vibrating thing. Put finger on vibrating thing and go bzzz, and then my finger starts being numb because I've vortexed it too long. How can anyone resist vortexing their finger when it's sitting right there? It's sitting right there. I love that. I would buy one. I would buy one just for that. And I know you're gonna say like, hey, like you can accomplish that. You can accomplish that through other means, but no, it needs to specifically be this blue box vortex machine. That doesn't sound good for the finger. No, it's probably not, but it's kind of fun. I love it so much. If I can have one, a goal would be to own one and to vortex my own finger whenever. I wouldn't actually vortex my finger all the time, but it would just be fun to have. It isn't. That's the reason why it's a legit occupational industry for people. Yeah, it's really not. That kind of vibration, especially like I was very mindful not to be at like the max setting. It would be at like the lighter settings. But it's just kind of fun. Oh, Western blots. Oh, Western blots. Ugh. I have such mixed feelings about Western blots. So if you don't know what a Western blot is, it's a technique you can use to detect proteins within a sample. So usually you separate the proteins through gel electrophoresis, you transfer them to a membrane, and this usually requires like specific machine that has the gel slowly move into like the membrane, and it's like electrical transferring, and then you have this membrane that has your proteins just in it. you have to now like wash this membrane with antibodies that bind to the proteins you are interested in, and that takes a while. You have to like it's literally like you put it in like a little container with some milk and your antibody, and you have it like rock back and forth for like an hour or two, and you keep doing that for like over the course of like, I don't know, like 2 hours. Depends on like it depends on your specific protocol. Some people have like overnight procedures for this. Anyway, overall, it shouldn't be too it in theory it shouldn't be too painful, because a lot of it is not so bad. It's a lot of waiting. So you're like, what's wrong with that? It's not that bad. And I'll agree with you. It's not that bad. Over the course of too long, yeah, it is way too long sometimes. I enjoy having overnight procedures with it, because you just throw it in the fridge and you're done. That's it. Good, cool. I like that. That's cool. But, and this is like might be specific to my grad school lab, because when I did this in undergrad, we had a nice machine to help detect, like to read our Western blots. We had a nice Western blot detector. But in my grad school, we didn't have a nice machine for that. And so what we would do is like we would have to buy like x-ray films and do like darkroom x-ray like detection methods. So when you like put the antibodies on, they do a bit of like they have a ability to kind of like chemo illuminate, like when you like put a specific chemical on them. And so you would get these very sensitive x-ray films and then like put them on top of the membrane to like make the luminescence go onto the x-ray film. And so I absolutely hated being in the dark room and doing all this. I cannot explain to you how much I would have like a mental breakdown when it was like time to do a Western blot and I had to be in the dark room for like an hour. Like, I can't see anything. I'm like, and the room is really big, and I'm feeling my way around like the table, going like, okay, there's my like membrane, there's my x-ray film sheets. Okay, let me do that. Let me make sure they're sandwiched together, and I have to wait, and I have to do a timer, but I can't use a timer because I can't use light in the dark room, or else it might affect my x-ray film. And so I'm like counting in my head, going like 1, 2, 3, 4, 5, 6, 7. And then after you do that, you have to slowly make your way to the x-ray like detection machine and like feed your x-ray film into this machine. And I was absolutely losing it. It was like insane to me, like that I had to do any of that, because I just remember going like, oh my gosh, there's machines for this. Why am I doing this in this stupid dark room? I hated it. This is definitely like a implode. If I didn't have to do any of that x-ray like detection, like I probably would enjoy. This would be higher up. It like it breaks your brain. It really breaks your like brain being in the dark room for like 2 hours. We had a dark room at my old lab, and yeah, it was never used by us, but I've seen people go in and out. I was never experienced with it until like my thesis lab, and I was like floored. Also, I think the person that was teaching me really, really did not. I don't think she was like enjoying the fact that I didn't like it. It's not like I was making it known. I was trying to be obviously as pleasant as I could be when I'm being taught by someone like a stranger. And but I was like kind of like struggling. Like I was going, oh, I don't like. I bumped into her in the dark room. I'm like, oh, I'm sorry. I'm really struggling here, trying to figure out my way around the room. And she's like, I'm like, I'm sorry. I don't know. Makes you wish you took dark room photography in high school if they offered it. Yeah, maybe if I like, maybe if I like had that experience before, it wouldn't be so bad. But overall, it was just like, oh my gosh, I hated it. Cell culturing. Okay, my experience with cell culturing is actually quite limited. I put this here because I feel like I can't call myself a bench scientist if I didn't like talk about cell culturing a bit. But I only did this for like, I want to say like a year, if that. I love okay, I love forbidden fruit juice. It is that's like the biggest upside of this was I was like I feel like the media looks so good for like I want to drink it so bad, but you can't. It definitely it was like more aesthetically pleasing than bacteria culture. Bacteria culture is usually like just yellows and grays, greens and like kind of ugly colors. It was so colorful in the cell culture incubator. You've tasted it, E-Man, before? I'm guessing it's just salty. Isn't it just like mostly salt, like salt water? Tasted like saline. Interesting. Yeah, that's about what I figured. I kind of want to try it. Maybe one day on stream. Maybe one day on stream we'll have a culture lab. RPMI is sweet though. Interesting. Maybe we can have a taste tier list, but don't replicate this at home, kids. Cell culture, I'm gonna put this in I am neutral simply due to the fact I feel like I don't have enough information to say otherwise. I think I would hate this. I would think I think I wouldn't like this, but from my experience with it, it was not so bad. I think because I'm comparing it to mice, which were quite needy. They were like needy and I had to come in the weekends, so people always say like, oh, cell cultures are so like you have to go visit, you have to go like you have to check on them so often. But I'm like, well, I worked with mice and like I had to check on them even on the weekends, especially when I'm doing certain like experimental protocols. It wasn't so bad. It's fun until you get your cultures contaminated by mycoplasma. Yeah, I have heard like contamination is not very fun, but again, I think I'm neutral. Leaning towards I would probably not be a big fan. Elisa's. This is crying in the morning tier. Cell culture becomes exponentially worse if you do virus work, though. Feeding isn't that bad if you schedule their feeding. Oh yeah, I never did like anything related to viruses, but I have heard that it's very unpleasant. I was basically scared off of working in like any virus related lab because of like the horror stories I've heard. If you're doing a growth curve, you're basically sleeping in the break room. Yeah, okay. Yeah, I'd probably not like it. It's okay. It's okay. Elisa's are definitely in the crying in the morning tier. I think because I don't think I've ever gotten any of my Elisas in any point in my career to work. Like, I feel like every Eliza I've done has flopped. If you don't know what an ELISA is, it's enzyme-linked immunosorbent assay. And so you usually use this to detect and measure how much protein or antigen or antibody there is. And so it's like I would say pretty qualitative. I would consider it qualitative because it's just like based on color changes. Though you use like a machine to kind of measure like the amount of color change. So it makes it a bit more quantitative in that sense. But it's the darker color equal more protein there. I don't think this ever worked for me. And I don't know what I was doing wrong. I only did this like a few times, but I only did this a few times because it kept failing. But every time I did it, my controls never quite lined up. I think my pipetting was just bad. I think I just content. I think my pipetting was just bad. That's all I can say about it. Like, I feel like you would have a control so you can tell, like, this is this much concentration, this is this much, and it just I don't know. It's I was not meant to do these things. I don't know what to say. I couldn't. I'm sure it worked for us, but that was my intro to biology and also the work that was done by the commercial COVID test kit. Yeah, I'm sure. No, definitely. I'm sure it works for people. In my lab, I've seen people do it phenomenally. I just suck at it, I think. I think I really struggled with anything with precise pipetting. I think something like I'm not very good at pipetting. I say I enjoyed pipetting, but maybe I'm not good at it. Okay, I'm gonna do the last experiment. I think you picked soft that Elisa got and decided to curse you. Yeah, maybe Elisa herself was upset at me. SDS page. This is like basically gel electrophoresis. This is basically gel electrophoresis. I would say I liked this better. It's not ranked in tiers, but this was a better gel electrophoresis because of the fact I didn't ever have to make the thing. You just, you open up a pack. You open up the gel pack, the pre-made cast gels, and that was it. And yeah, there's SDS. I didn't really mind the SDS. Yeah, we just bought our gels. I think I would hate it more if I had to cast them. Because I know I casted them in like some labs, but it was really brief. But for when I had to do like a lot of them, like when I was doing like an SDS page like a week, we just had pre-made ones. But just because it'd be way too annoying to do it. The amount of times I had to like, yeah, if this was like manually doing it, this is definitely like I would go down a tier because the amount of times your stupid little gel would like leak. It was so bad. So dumb. Anyway, it's not the worst. I enjoyed it. I enjoyed it because I did a simplified version of it and I also didn't have faulty equipment where things were leaky. When things are faulty and leaky, that is the worst that can ever happen with an SDS page. When things don't work because your buffers are leaking in the wrong area, that's not fun. That's really bad, and I will cry in the morning because things are leaking all over the place and you just wasted so much time. But because it was all good and like working, we put this in. I enjoyed it and did not dread waking up in the morning to do the SDS pages. And now we've finished all the lab experiment parts. And now we're going to go into things that are science adjacent. Yay, science reading and writing. I love those things so much. Oh God, give me more science reading and writing. That's so good. I'll never forget being told, like, oh my gosh, how dare you keep reading papers? Go do something. Go do something. I understand why I understand why people were mad that I was just kept reading papers instead of doing lab work. I get it. They pay me to go do lab work, not read papers. I like reading slightly less than writing. I curse the gods for giving me not ADHD. I think it's hard to say which one I like more. I think reading is a lot more interesting. But writing has like its own kind of fun related to it, especially depending on what kind of writing you're doing. When you're writing about your own research and you're doing like an academic like paper, I think writing is a little less fun for me because it's so strict in what you're like it's so strict what you're writing about, and like there's specific styles you have to look for, and also I think sometimes also word limits are kind of annoying. It is, but not gonna lie, after 2 sentences my eyes just can't focus on the words anymore. Yeah, I get it. Like even like when I like my job nowadays is primarily reading and writing, and even when I'm like even when I'm reading papers now, I feel like I'm not like you're not really reading all of what's being written. It's a lot of like skimming with purpose. There's so many things. It sucks. Like, it's crazy how much we have to write as scientists in a paper, and then how much probably gets skimmed over. Who, like, most people are probably not reading the methods section, unfortunately, until you are the scientist doing the right, like, the methods section. AKA all of it, yeah. I remember when we were writing a grant, and my PI would tell me, like, listen, you have to, like, write very, very succinctly, because these people who are reviewing your grant, they're gonna, like, skim, like, 99% of it, and you need to make sure, like, at least one sentence captures all the information that they need. I was like, oh, okay. Grant writing especially. I like grant writing because I think it's a fun skill to perfect. And I've been doing a lot of that as of late as a science writer, because we need funding. that one I understand why people hate grant writing, though. It's so like, it's like everything bad about academic writing. Grant writing scares me. I can do reports and papers. It's a skill. It's a skill that you really have to practice. It's like incredibly distilled academic, like, paper writing. Like, you're writing for a journal, but you, like, are taking all the most important pieces of information and then also spinning it to, please, I need money, please, give me the money. I cannot for the life of me sell my research and myself as an asset. I think that might be the reason why I got fricked over for the grad school admission this year. Didn't do enough to say that I am a good pick. Please pick me. Yeah, it's really hard. Honestly, that was like, it was a class, or not a class, but it was like a seminar I like took, where basically the writing department was like, you need to learn how to sell yourself. And they're like, scientists don't know how to sell themselves very well. You have to sell yourself. You have to like make yourself sound like your research is the most important, bestest research of all, and they need to pick you. I'm like, oh, okay. Too pessimistic. Yeah, you really have to like put on like the salesman act as a scientist, especially if you ever want to be your own, like, you want, if you ever want to be the head of your lab. I think that was something I don't know if I would be able to do like a good job doing that constantly. Right now, I only write grants like sporadically, and I feel like it's a lot easier when you're writing grants about something that's not like, I don't know how to explain it, but it's not like exactly research based. I just want to do funny experiment. I know, just fund me in my funny experiment. Thank you. I'm worth it. And I think that's everything. 2 hours and 40 minutes. That's not so bad. I'm glad I didn't do more. I was gonna like, I thought it was gonna be too short, but it ended up yapping just the perfect amount. I think there's way more I could even talk about. Maybe I'll have a part 2 eventually, where I finally think about things that I did in other labs. Because most of this, again, was like in my most recent lab. I would have to like really sit down and think about how, like, what was I doing in these other labs? My brain truly has blacked out most of this information. Trauma blackout. Fungus lab. Yeah, like, I feel like I didn't get to do too much about like when I worked with pneumocystis. But again, I don't remember a whole lot of it. It's also worth recreating experiments to verify them, but that isn't profitable enough for funding, I guess. No, it's really not. It's crazy like what gets passed over for funding. they're looking for things. They're looking for things that are very marketable, which is sad. And that ends up we lose a lot of like basic biological research because of that. They want to see something that has like immediate clinical translation. I know this is a big ask, but once it comes to application, could you read over my letter? Oh, of course. I am super happy to do that. You, my DMs are always open. That includes like that goes out to like anyone in my community. I'm super happy to read over these things. Just let me know. Sorry, I'm eating a mango. I want anyone to get into the like grad schools of their dreams. I'm I'm pretty good, I will say, at doing these things. I used to like volunteer to help people with their grad school applications. Can't suss out false information is actually never verified. Very true. Uh oh, I wanna. Oh, that might be. Maybe that'll be the next Science Friday stream. I know I keep putting off the adipose lesson, but I think sometimes some of these things are more fun. I want to go through the redacted papers website. There's like a website where people like call out like bad science, and like it's like in a forum. It's a pub. I can't remember the name off the top of my head. Yeah, retraction watch. Yeah, retraction watch. I love it. I think it'd be fun to go over it. Some of it is like, it's insane. The level of fakeness that gets like through peer review. They are like, just copy and pasting figures left and right and going like, hello, how did no one catch this? Anyway, that might be fun. I might put that on the schedule for next week. It would also be easy. I also like doing these tier lists. I might do more of these for random science things. It's kind of just an easy way to yap. I realize I have a lot to yap about for each thing. If you do watch parties, Bobby Broccoli's videos, oh, they are pretty good. Um, those are really fun. Maybe I'll do some of those too. We have a lot of options. Science Friday will never be bereft of content. I'll have you guys know. Science Friday will Science friYAY will live on forever because my brain there's so many different fun science things. Yeah, I have seen those yeah, they're so cool. I think they're really funny. Okay. I think we're going to wrap it up here. Thank you so much for hanging out with me in my little tier list stream. I appreciate all of you, and I don't have any more streams this weekend, but I'll have a schedule up. I'll have a schedule up Sunday, I think. And we should have more streams. Probably more. I'll have some gaming streams next week. I didn't really game at all this week. But I will see you guys. And I'll always, of course, I'm on Discord and Twitter, and you'll see me there. Okay. Bye 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 bye. Bye, Weetabix. Yeah, have a good weekend, all of you guys. Bye.